Hi everyone, it's Roger Batchelor, and this week we're going to be talking about the chemical mind, and I'm also going to address some issues related to the textbook, which I'll do at the end of this lecture. And we're going to be dealing with this again next week. This is just an introduction to this biopsychology, and I've broken it up into the nerves and the nervous system and everything else because this is the more difficult part to understand. All right, so let's get into it. So when we're talking about the nervous system, we're talking about basically neurons, which make up the nerves. And the nerves are basically large bundles of axons, and we're going to, in a second, talk about what those are. We have the central nervous system, which is the spinal cord and the brain. And then we have the peripheral nervous system, which is basically everything else that's around that. So the peripheral nervous system is broken down into both the somatic nervous system, which controls our ability to move our body, and the autonomic nervous system, which controls basically automatic functions. So that's a good way to remember it. Autonomic covers automatic things, and somatic covers voluntary things. So the automatic nervous system is broken into the sympathetic and parasympathetic nervous system, which we'll talk a lot more about in a few minutes. So this is the part that we're going to spend quite a bit of time on, and that is that how the nerves are set up and how they work. So we have dendrites on one side of the neuron, which is a nerve cell, and then we have the soma, which is the stuff inside, and then we have an axon, which is the tube that connects it to the axon terminal, which is on the other end. So here's what it looks like, right? So the soma is basically the cell body. The axon, you can see here, actually follows through the myelin sheath and then ends on the axon terminals, which are connected to the dendrites of another nerve. So what's of the axon terminals? What you will see in the on this end of the cell, let me see if you can see yes you can. So what you can see on this end here, that is what's connecting here to the next cell. So the axon terminals then connect to the dendrites on the next cell. Okay? And this is the axon that's basically this long piece that's going to the end of the axon terminals. And we have an impulse happening in here. So the nerves are actually sending an electrical signal, right? So up above here in this picture, you can see a nerve cell in cross-section, and it's basically under a microscope, so you can see what it might look like. The nerves travel from the dendrites, as I mentioned, to the soma, which is the cell body, and then to the other end of the axon, right? So this is a motor neuron that's shown, and recognize that these neurons stretch from the brain and spinal cord to the muscles of the body. And we're going to talk more about these when we get into the reflex arc. So how does this all work? Well, that's actually electrical signal that's passed, right? So the resting potential inside the nerve cell is about minus 70 millivolts, and the threshold to basically trigger a reaction is minus 50 millivolts. And so what happens is the cell is in a normal resting potential, meaning there's nothing happening. It's at minus 70. And then when it gets to minus 70 with the outside, then it triggers a reaction within the nerve cell. And basically the sodium ions, which are positively charged, run into the cell, and then the charge within the cell, just for a brief millisecond, becomes positive, and then the positive potassium ions then flow out of the axon, and it goes back to being negative. So we have these ion channels, which are on, these are on the, let's go back to the nerve here, the nerve cell. The ion channels are actually here along the axon. And they're tiny, tiny, tiny little holes that let the, as we mentioned before, we have sodium, rushing in and potassium rushing out. And they need somewhere to do that, and that is the ion channels. And so, although it's positive, it becomes negative afterwards, right? And the synapse is that little gap, that little tiny gap, and I'll show you here. It's that gap that connects the dendrite to the axon terminal. And we're going to get a closer look at it in a couple of minutes. And the myelin sheath, this is the sheath that extends along the axon. And the reason that it's there, it actually speeds up the transmission of the signal. Okay? And then saltatory conduction is what basically we were talking about, is the fact that this is basically a salt, right? So sodium is a salt and potassium is a salt. So here's a good video that will take us through the beginning of the how does the nerve cell work. It's a very simple one. I started with this. We've got more coming. This is a good one to start with. Here we go.
How do nerves work? Are nerves simply the wires in the body that conduct electricity, like the wires in the walls of your home or in your computer? This is an analogy often made, but the reality is that nerves have a much more complex job in the body. They are not just the wires, but the cells that are the sensors, detectors of the external and internal world, the transducers that convert information to electrical impulses, the wires that transmit these impulses, the transistors that gate the information and turn up or down the volume, and finally, the activators that take that information and cause it to have an effect on other organs. Consider this. Your mother gently strokes your forearm and you react with pleasure. Or a spider crawls on your forearm and you startle and slap it off. Or you brush your forearm against a hot rack while removing a cake from the oven and you immediately recoil. Light touch produces pleasure, fear, or pain. How can one kind of cell have so many functions? Nerves are in fact bundles of cells called neurons, and each of these neurons is highly specialized to carry nerve impulses, their form of electricity, in response to only one kind of stimulus, and in only one direction. The nerve impulse starts with a receptor, a specialized part of each nerve where the electrical impulse begins. One nerve's receptor might be a thermal receptor, designed only to respond to a rapid increase in temperature. Another receptor type is attached to the hairs of the forearm, detecting movement of those hairs, such as when a spider crawls on your skin. Yet another kind of neuron is low-threshold mechanoreceptor, activated by light touch. Each of these neurons then carry their specific information, pain, warning, pleasure, and that information is projected to specific areas of the brain, and that is the electrical impulse. The inside of a nerve is a fluid that is very rich in the ion potassium. It is 20 times higher than in the fluid outside the nerve, while that outside fluid has 10 times more sodium than the inside of the nerve. This imbalance between sodium outside and potassium inside the cell results in the inside of the nerve having a negative electrical charge relative to the outside of the nerve, about equal to minus 70 or minus 80 millivolts. This is called the nerve's resting potential. But in response to that stimulus, the nerve is designed to detect pores in the cell wall near the receptor of the cell open. These pores are specialized protein channels that are designed to let sodium rush into the nerve. The sodium ions rush down their concentration gradient, and when they do, the inside of the nerve becomes more positively charged, about plus 40 millivolts. While this happens initially in the nerve right around the receptor, if the change in the nerve's electrical charge is great enough, if it reaches what is called threshold, the nearby sodium ion channels open, and then the ones nearby those, and so on and so forth, so that the positivity spreads along the nerve's membrane to the nerve's cell body, and then along the nerve's long, thread-like extension, the axon. Meanwhile, the potassium ion channels open, the potassium rushes out of the nerve, and the membrane voltage returns to normal, actually overshooting it a bit. And during this overshoot, the nerve is resistant to further depolarization. It is refractory, which prevents the nerve electrical impulse from traveling backwards. Then, ion pumps pump the sodium back out of the nerve and the potassium back into the nerve, restoring the nerve to its normal resting state. The end of the nerve, the end of the axon, communicates with the nerve's target. This target will be other nerves in the specialized area of the spinal cord to be processed and then transmitted up to the brain. Or the nerve's target may be another organ, such as a muscle. When the electrical impulse reaches the end of the nerve, small vesicles or packets containing chemical neurotransmitters are released by the nerve and rapidly interact with the nerve's target. This process is called synaptic transmission, because the connection between the nerve and the next object in the chain is called a synapse. And it is here, in the synapse, that the neuron's electrical information can be modulated, amplified, blocked altogether, or translated to another informational process.
So hopefully that was useful. I think it's a nice and simple introduction to how the nerves work, and we're going to be talking more about this in just a minute, once I can get this to go to the next slide. Here we go. So as you look here, as I mentioned in the video, the axon has a negative charged interior in the resting state, and then with the action potential, the ion channels open up, and the ions rush into the axon to try to neutralize that negative charge, right? So the positive ion rush in, and then the electrical charge moves from negative to positive inside. And then as the action potential is moving along the axon, positive and negative charges are reversing, right? So it goes from negative to positive as the ion channels open up, and then after the action potential passes, the positive ions flow out of the axon to restore the negative charge, right? So it goes back to how it was. So this is showing the ion channels as I was talking about. There's the sodium. If you see at the top, the sodium here is rushing in, right? Both from top and bottom, and that's making it from negative to positively charged. And then it goes along a sequence here, right? And it would travel from left to right, looking at this picture here. And after it's passed, then the potassium flows out of the axon, which creates the newly negatively charged ion. And then eventually it gets restored to the original resting potential. So this video, once again, will talk more specifically about this action potential.
What if everything you did, thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between turn up the thermostat and oh my gosh, I'm on fire. That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we've got to first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all or nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon, because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That You will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale. The script was edited by Blake T. Festino, and our consultant is Dr. Brandon Jackson. It was directed...
Alright, so that gives you a good idea about how the nervous system works. And so let me just go ahead to the next slide, and we'll talk about how this actual transmission takes place. So at the end of the axons, they have these vesicles. And basically the end of the axon, which is called the axon terminal, it's connecting but not touching the dendrite in the next nerve. And as this action potential moves along, it releases these little sacs which have the neurotransmitters in them. And those sacs will then attach to the proper sites on the other end of the on the dendrite. So the axon terminal has the sacs. It releases the sacs, which releases the neurotransmitter. That neurotransmitter passes across the very tiny synaptic gap, and then attaches at the proper receptor site on the other side. All right, so. This shows you, I mean, this is really, really exaggerated. It's actually only 0.00025 millimeters, right? And some of these neurotransmitters actually excite the next neuron, and some neurotransmitters actually unexcite or inhibit the next neuron. So, dopamine and acetylcholine are two of these neurotransmitters, but as I mentioned before, there's actually many of them. So here's some of them. So in terms of excitatory, acetylcholine and dopamine, GABA, which is inhibitory, glutamate, which is excitatory, norepinephrine, excitatory, and serotonin is one that inhibits, right? So in terms of what it does in the brain, acetylcholine participates in the movement, learning, and memory. Dopamine in motivation, reward, planning, and behavior. GABA inhibits the central nervous system and is involved in mood. The glutamate has an excitatory effect in the CNS, and norepinephrine participates in arousal and mood, and serotonin participates in mood, appetite, and sleep. So here's another video that talks more about this synaptic transmission.
In the last module, we took first steps towards understanding the electrical properties of individual neurons. We learned how electrical forces and diffusion give rise to membrane potentials, and we learned how cells can generate and propagate signals called action potentials, or spikes, along the membrane. Understanding the properties of the neuronal membrane is essential, but understanding just these properties isn't sufficient to give us insight into collective behavior of the billions of connected neurons in our brains. Luckily for us, we can approach neuroscience at many different scales and levels of analysis, and we don't have to confront the full complexity of everything all at once. That's what we'll be exploring throughout the rest of this course as we slowly go from our understanding of single molecules, such as ion channels, to the electrical behavior of neurons, to their collective behavior in small circuits, and finally onto how they become organized in large functional regions of the brain. Let's start simple though. Since we've examined one neuron, a logical next question is how do two neurons connect with one another? We'll first examine some basic cellular anatomy of neurons. So far, we haven't made too much of the fact that the majority of neurons are polarized cells. That is, they have one portion of the cell for receiving inputs and another portion for sending outputs. The parts of the cells that are specialized for receiving inputs from other cells are called dendrites. The word dendrite comes from the Greek word dendron, meaning tree, and as you can see, the dendrites have a branching, tree-like structure. a signal received by dendrite is passed to the cell body. If there is sufficient depolarization of the cell body membrane to initiate an action potential, then an action potential is sent down the axon. The axon then carries the propagating action potential to another neuron. So what actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another? This interface is called a synapse. There are two general types of synapses: electrical synapses and chemical synapses. Electrical synapses are less common in our own nervous systems, but they're simpler to think about, so let's start with them. Electrical synapses are basically pores between two cells that allow ions to pass through. They allow the passage of the electrical signal through to a neighboring cell without much fuss. It's not so different than just combining two cells into one larger cell. There are lots of reasons that nature might need synapses like this from time to time. They're fast and they allow cells to couple together with a high degree of synchronicity. But most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior. We'll also look at the role of defective synaptic physiology in neurological and psychiatric disorders, and we'll see how synapses can be targeted by various psychoactive drugs and poisons. Finally, we'll wrap up by looking at how synapses can change over time in response to external stimuli, playing a foundational role in how we learn and remember.
Yeah, that was great. I really appreciate the fact that that was done by Harvard. They really did a good job, right? So the next thing we're going to talk about is neural networks, and recognize that the nerves actually work in bundles in these networks. And it's like a computer network. You know, think of a computer network, all the computers are connected together. Well, here we have all the nerves connected together to do something, right? Because each nerve is receiving as they talked about, this excitatory inhibitory message, but they're all working together at once. And part of this neural network is the reflex arc. So the reflex arc basically is when we have something negative that the nerve is experiencing, and instead of having to send that pain back up to the brain to have it all computed to figure out what's going on, what happens here is that message just goes to our spinal cord and then goes directly to the muscle to remove the part of the body that's experiencing the pain, right? So here it's showing a stimulus to the skin on the foot. It's going up into the spinal cord and directly back into the leg, the muscle in the leg, to remove our foot off of that. Of course, this happens very, very fast. But it'd be much slower if we had to go and actually think about doing it, right? And that gives us a protection from things that might be noxious in our environment. So here's a great explanation of the reflex arc.
Hi everybody, welcome back to Miss Angler's biology class. I am Miss Angler. In today's video, we are going to be looking at a reflex arc, and I'm going to be walking you through how the impulse travels through your spinal cord and how decisions are made when we speak about reflexes. It's also at this point, if you're not so familiar with structures of the nervous system, that you should go click the link above now to go watch an introduction to the nervous system where I walk you through the basic structures that you see today in this picture. I'm also going to run through the significance of a reflex arc, as you need to know why we actually have this particular structure. Now, if you are new here, don't forget to give this video a thumbs up, subscribe, and turn notifications on, because I post new videos every Tuesday and Thursday for grade 8 to 12 biology. And as a little extra, if you are in matric and you need that little extra help, you should think about joining my membership, where I upload my amazing summaries. I do a lot more exam prep, and I walk you through exam papers, as well as having live lessons with me. So we're going to start our journey with reflex arcs, starting at the point that you experience the stimulus. Now, you are generally going to experience your stimulus through your skin, through your eyes, maybe through your ears. But in this example, I'm just going to use your skin. And these are a few of your surface skin cells. And located inside of these skin cells are going to be receptors of some kind. So I'm just going to draw those in red. And there's one inside of each of them, and they're all connected to each other. And so these receptors, which are in your skin, are going to collect the information they're receiving. And let's say, for this example, you've put your hand down on a hot stove. Now, that information is going from those receptors, and it's going to travel down a neuron, and that neuron is going to enter into your spinal cord via the dorsal root of your spinal cord, and it's going to make its way towards the gray matter. Now, this particular neuron, you're going to need to know this, is a sensory neuron. And it's a sensory neuron. We know this because it's obviously bringing in the sensory experience, what you're feeling. We know it's also a sensory neuron because its cell body, which is this little thing I've drawn on the side here, this circle, is off to the side of it. Now, once that information comes into the spinal cord, it now needs to be transported, into the actual spinal cord, because the spinal cord is actually going to make the decision, not the brain. And so we need a specialized neuron, and this specific specialized neuron sits in the gray matter, the full length of it like that. And now an integrative measure needs to be taken, and so we call these neurons either connector or interneurons. And this is where we'll make our decision. So our spinal cord says, we are putting our hand on a hot plate, we are hurting ourselves, we need to move. So it makes a decision. We're going to move, and that's the choice. Now, it sends that particular message to the next neuron, which sits on the other side of the spinal cord, and that neuron is our motor neuron, and it makes a little synaptic join with our connector, and it runs the other side, or the dorsal side of our spinal cord and the spinal root, and it goes out and towards its destination. And so that's our motor neuron. It's going to cause the movement. Now, the motor neuron itself is attached or embedded in what we call an effector. And in this picture here, I'm just drawing a muscle, a striated one we would get in our skeleton. And this particular muscle is going to help us move our hand away from the hot object. And so we call this muscle a effector, and muscles are examples of this. Glands are responsible as well for being effectors. And you can see the motor neuron sits inside the muscle. It causes the muscle to contract, and then you pull your hand away very quickly. Now, in an exam, you need to be able to explain what I have just said now, and it's generally a describing question. You're going to describe a reflex arc and how it works. Another thing they may ask you is the significance of a reflex arc, like why does it happen? And the reason is we need response quickly to any kind of safety issues as well. And that response is quick because it goes from the stimulus to the spinal cord, and then you respond quickly. You move, or and some of the examples I'll run through now, you'll like blink. And that's because we can bypass the brain. The brain doesn't have to make any decisions. The spinal cord makes all the decisions for us. And this is also sometimes when you burn yourself on a hot plate. You actually move before you feel the pain. It's these split seconds where you didn't actually know you were burning yourself, but your spinal cord already knew that you were hurting yourself, and so you quickly move away. Now, some of these examples that I've listed here, like coughing, yawning, and blinking, they're not just the regular coughing and yawning and blinking. These are all safety measures. And what I mean by that is they are safety measures to prevent you from like dying. So things like coughing, something stuck in your trachea, the reflex to get that out is coughing. If there's too much carbon dioxide in your blood, you yawn more so you get more oxygen in. And likewise with blinking, when somebody throws something at your face too quickly or you hear a loud sound, you generally blink as a way to protect your eyes, because they're actually one of the most important receptor organs that humans have. So we want to keep those safe. And that's another way that they can ask this particular question. The last thing they can probably do is ask you to draw a reflex arc. Now, as always, I like to finish off my lessons with a terminology recap. You can use these words for flashcards, and terminology is really important in life sciences, so you need to know how to use these words properly. The first thing we're going to look at was the reflex arc, which is the actual pathway itself where the electrical impulse runs from the receptor into your spinal cord and then out to the effector. That is the arc itself. Then we looked at receptors, which are essentially these specialized cells that experience pain, pressure, temperature. They tell you information from the outside of the body. And it runs along the sensory neuron, which is the neuron that carries that sensation. It then goes into the spinal cord where we find the interneuron, also known as a connector neuron. And that neuron is where we make decisions. That decision is then sent via the motor neuron, which is a moving neuron. It means it's going to make things move. And that is sent to the effector, which in this case was a muscle, but it can also be a gland. These are the structures that carry out the response. That's what an effector is. And we finally spoke about reflex actions. And the actions were those examples where I said things like blinking, sneezing, coughing. That's the action that's brought about. Now, as always, if you've liked this video, don't forget to give it a thumbs up and subscribe, and I will see you all again soon. Bye!
All right, everyone, so I think we understand about the reflex arc now. And we're going to go back to the neural networks. Here we're talking about the fact that we're showing a small neural network. And of course it's much more complicated than this, right? But the idea here is that the A neuron at the top is receiving input from two weaker neurons, right? And it combines these and then two other ones, and it's combining this information into a decision whether or not to launch the action potential, which we've already talked about, right? Now, to end off, just to talk a little bit about neuroplasticity, and that's basically the fact that our brains can really, really change to deal with whatever it's whatever it's dealing with. So, if we have a head trauma, then it can build itself up, it can shut areas down, it can get into the areas to move faster, and these neural networks we talked about are constantly in a changing mode, and the fact that our brain can really adapt to whatever environment that we're in. There's work that's been done to repair a damaged brain. They know now that the brain will actually repair itself, which is neurogenesis, but they're doing some work with stem cells and other treatments to try to get the brain to work after a traumatic brain injury. And that's essentially it for this part. I mean, I just wanted you to get more familiar with how the nerve is working and some of the chemicals involved. And I did promise you that I was going to talk about this textbook now. So because so many people complained about having to buy the textbook, which I find very strange, but nevertheless, I've been asked by administration to make alternative arrangements rather than you having to do the assignments through the textbook. So what I will do is I will create some assignments for the people who don't have the textbook. Actually, they'll be for everybody. But they will be open book. If you have the textbook, you'll be able to go a lot faster, obviously. They're going to be little short tests that are very, very done in a very short period of time. So it means that you won't have time to do much if you don't know the answer. So I'm going to give you like probably around between 20 and 30 seconds per answer. So it's testing whether or not you actually have the knowledge in your head rather than whether or not you can look it up online. All right, so stay tuned. I'll have those ready within the next couple of days and let you know more about that when it's ready. All right, I'll see you for the next one. Bye for now.